In this third and the final video in cytology section, we're going to talk about nucleus. Earlier, nucleus has been considered by some physiologists an organelle, a little bit larger than anything else that exists in the cell. But today's trend is to present it as the nucleus, not to make it fall into a category of organelles, because its role is quite uniquely different, and as such, clearly deserves a section on its own. In this illustration, as we can see, a nucleus, which is quite significantly larger than any other recognizable structure within the cell, is next to or practically closed in the center, created by rough endoplasmic reticulum. If we take a closer look at the nucleus, we're going to see that in its core exists a nucleolus, which means a small nucleus, surrounded by amorphous mass, which is called the chromatin. But we know the chromatin is just dissolved DNA, which, when the cell is ready to go through a process of cellular division or mitosis, will start twisting and coiling more tightly upon itself to form a number of chromosomes that during the cellular division become visible as individual units. And for human cells, we should expect to have 46 chromosomes within all the human cells, with the exception of reproductive cells that would have just 23, whether it's a male or a female reproductive cell. Because the content of the nucleus is so precious, it cannot be allowed to accidentally diffuse into the rest of the cytoplasm. So we're going to have the nucleus also exhibiting its own nuclear membrane, where the red markings all over the membrane itself are actually the nuclear pores, openings that are big enough to allow a molecule of messenger RNA, once it becomes formed, to emerge safely out of the nucleus, while the DNA, regardless within which stage it is found, whether it's in the stage of chromatin or in the stage of chromosomes, simply it's going to be too large, too massive to be allowed to emerge out of the nucleus through the nuclear pores. 8,000 time enlargement electronic microscope of a nucleus at the bottom of the screen again shows some kind of amorphous mass, which was given the name of chromatin because early physiologists or cytologists have noticed that the content of the nucleus dyes differently compared to the rest of the cell when submerged to some kind of special dyeing process. And Greek word chromos comes from color. So they didn't know what the structure is, but they were just able to understand that it absorbs the dye, and they gave it a very neutral name, chromatin. Years would pass, better to say centuries, until 1950, and Watson and Crick gave us the first model of what they thought would be interesting for science, the model of DNA as we recognize it today. Nuclear envelope or nuclear membrane is also quite clearly visible on this photomicrograph, including the nuclear pores that are present throughout this picture at the multiple locations. So this is a little bit of interesting involvement of how the DNA actually becomes recognized. We already know from section that was titled Essentials of the Organic Chemistry that individual nucleotide containing one of the 4 nitrogenous bases, molecule of deoxyribose, the pentose sugar, and a phosphate group are essentially to be found within a nucleotide, and then placing nucleotides in chain and allowing to form 2 helices and establishing hydrogen bonds between complementary bases, such as adenine to thymine or thymine to adenine, and the other pair that could be generated as cytosine to guanine or guanine to cytosine, enables 2 helices of the DNA to stay connected they would disconnect when we have need for duplicating the DNA, as it's happening in a period of time when a cell is preparing for its replication, for division, mitosis, or we would allow just part of the DNA to separate its two helices to allow us to copy them to create a messenger RNA, and on the template of a messenger RNA, we would be able to export the message through the nuclear pore, make the contact between messenger RNA and ribosomes, and to observe process of protein synthesis. Both will be part of this presentation, but towards the end of it. DNA by itself would not be able to exist. It needs to use different proteins, they're called histones, around which the DNA wraps itself quite tightly. About 140 nitrogenous pairs, the nucleotides, are seen wrapping itself around a group of histones. And that is the segment of the DNA that then we recognize under a different word, calling it the nucleosome. Nucleosomes are linked to each other through a short strip of DNA that is called a linker DNA, so they appear to be some kind of beads on a string. And this kind of appearance of a DNA actually has led to invention of the term chromatin. Of course, if the DNA becomes even more tightly coiled upon itself, it appears to generate even more visible and larger and more tightly coiled molecule, which we then start seeing under better quality optic microscope, and we can recognize it as the chromatin fiber. However, further tightening, further twisting and gripping of the individual loops of a chromatin fiber will finally result in appearance of structures that could be not just recognized and made visible, but they could be even counted within a nucleus of a cell, which is undergoing cellular division. Of course, we're talking about chromosomes. You realize that the term chromo or prefix chromo appears to be present throughout this diagram, whether it's chromatin, chromatin fiber, chromosome, or chromatid individually. It is all because of the same consequence that the DNA actually was accepting a biological dye, and from that time, all the content has been referred to something that absorbs the dye. Total number of chromosomes within a human cell is 46, and they're arranged in 23 pairs. Why 23 pairs, not just 46? Because we are inheriting one chromosome within a pair from our mothers, and the other one within the same pair is inherited from our paternal side. So 23, which seemingly sound like we're duplicating same chromosome from mother, same chromosome from father, but they carry slightly different genetic characteristics. And that's why we're not clones of our parents, but rather we become some kind of interesting mix of some of the qualities that we have inherited through our genetical inheritance. A chromosome, which is illustrated here, does have a shape of a letter X, and as you can see, it is formed by two sister chromatids that are held together at the center point through section that is called the centromere. Basically, this shape of chromosomes is happening when a cell is duplicating its DNA content. When a cell separates, generating two daughter cells, link between sister chromatids at the centromere will be broken, and one sister chromatid will go into daughter cell number one, and the other one will be pulled into second daughter cell, so they would be separated. But as it stands right now, they're identical copies of each other. So two daughter cells will inherit from a mother cell which is undergoing cellular division, exactly the same genetic content. And this is important to understand that for somatic cells, cells of our body, if we're replacing older mother cell with the two younger ones, daughter cells, we want them to maintain the same physical and chemical qualities and characteristics as the mother cell had. Why? For example, our skin is a good example because it regenerates very quickly. Practically, we're getting new skin every 21 days. And our skin always looks the same. Should it not follow this principle, every time cells divide, we're going to be facing a new product, a new quality, a skin that would be more susceptible to one type of change, or a skin that will be more resilient to something else. But we would be dealing with a constant unknown. So because cells inherit identical genetical content from a mother cell, basically they can maintain the same function, same form, same shape, same physical or chemical characteristics as the maternal cell provided them. In a case of reproductive cells, when we're observing process of forming the reproductive cells, it is called cellular meiosis. Mitosis is division of somatic cells, but process called meiosis is the one which generates reproductive cells. Firstly, we would have to understand that from 46 chromosomes, we need to come down to 23. Because when two reproductive cells of 23 chromosomes each come together, they restore number 46. So 23 plus 23 equals 46. That's why I said earlier, we have 23 pairs of chromosomes that we inherited. Within a pair, a chromosome came from paternal, a chromosome, other one in a pair came from maternal side. Obviously, this has to be something different. So when we reduce the total number of chromosomes to 23, then through the second meiotic division, basically we would allow partitioning of sister chromatids, which have already been subject to some kind of free genetical exchange between themselves. And that's why amongst 4 daughter cells generated through a single meiotic cycle, basically they exhibit genetic variance. And we're not becoming clones of our predecessors, neither we would ever be able to see our own offspring being exact clones of us. So that's the role of DNA that we're going to observe in cellular division. Now, when we're talking about synthesis of proteins, then the nucleus practically assumes complete different meaning and completely different value. It becomes something that we can say the brain of the cell or processing unit or a microchip that is embedded into a cell, because it is going to allow temporary separation between 2 helices of a DNA, of which one of them will be transcribed, copied, but as a result of its copying, we're not getting another strand of the DNA, except it is going to be this time the RNA, so-called the messenger RNA. RNA differs from DNA in 2 different structures that we're going to find within a different individual nucleotide. One which is common for all is that we're going to have sugar, ribose, instead of deoxyribose that we observed in a DNA molecule. The second change is that the molecule of RNA would not have nucleotide with thymine as the nitrogenous base within it. It will be uracil, so adenine, thymine, adenine, cytosine, guanine, and instead of thymine, uracil. So if we're transcribing one of the 2 strands of the DNA and we're reading first nucleotide having adenine, first nucleotide that messenger RNA will form will be made of nucleotide with uracil. If the second one is T-thymine, messenger RNA would respond appropriately with adenine. cytosine matched with guanine, guanine matched with cytosine. So as a result of this process of transcription, we're going to see series of nucleotides with adenine, cytosine, guanine, and uracil being arranged, and a single strand of RNA molecule will be synthesized on the basis of transcription process of just one segment of the DNA's strand, not separating them and reading them from beginning to the end, just one strand. Different length, different proteins, of course, need to be produced. At this point, messenger RNA will emerge safely out of the nuclear space through the nuclear pore, and as the first door neighbor to nucleus is the rough endoplasmic reticulum, extremely rich with ribosomes. Small and large subunits of the ribosome will assemble and start reading the message encoded by a molecule of messenger RNA. On the basis of 3 different nucleotides, the ribosome will decipher this message and understand it as a set of 3 nucleotides that are coding for specific amino acid, which needs to be brought in. Transport RNA will be bringing individually amino acids, while the ribosome will ensure that they become linked in a chain in an appropriate sequence of amino acids until this process is over and newly formed protein is released further into the rest of the cytoplasm. Step number 2, decoding the messenger RNA and synthesizing sometimes short, sometimes long chain of proteins using amino acids is step 2, which is called translation. So transcribing from a language that we do not understand, but we can copy individual characters one by one. Later on, when we read the copied page, with the help of dictionary, we can translate. And as a result of this translation, we start getting a little bit of a meaning and understanding of what's going on. In this case, for a single cell, translation means setting an appropriate sequence of amino acids, enabling them to link themselves into chain through dehydration synthesis and forming shorter or longer chains that are now recognized as proteins. A little bit more about this process of transcription and translation. So as we can see here, we have the DNA, and of course, segment of a DNA is considered to carry significant information to form a single gene. At this point, I want to throw a very interesting number at you that with a complete reveal of a human genome, basically it has been proven that we are made of about 30,000 different genes that are found on our chromosomes. Compared to fruit fly, we're not too complex compared to it because genome of a fruit fly consists of about 18,000 genes. So we're just about 12,000 extra genes more complex than a fruit fly. Up until that point, the interesting thing was that scientists were thinking that we would have same number of genes as we would be able to identify individual proteins from a human body. And the proteins were identified to be somewhere in a range of about 100,000 different proteins. So conclusion was made that our genome is expected to have 100,000 plus different genes. It has been proven wrong hypothetical idea because many of the unknown factors at that time simply were not taken into consideration. Now, as we know a little more about the DNA, it is definitely making it easier for us to understand that certain segment of the DNA might be coding for multiple proteins. So one gene essentially could code for 1, 2, 3, or even more different proteins, just depending what kind of message is read, transcribed, and then copied onto messenger RNA and then translated into protein. Let's think about code being made of 4 letters. Letters are C, A, R, and T. If we say this is the entire code and we try to read it beginning to the end, we understand it. It's a word, cart. Now, what's going to happen if we delete one of the letters and instead of 4 have 3? Well, with the deletion of R, we're going to have CAT, equivalent of cat. With deletion of letter T, we're going to get car, CAR. With deletion of A, we're going to have CRT, abbreviation for court, as typically seen in our addresses. And even without letter C, we're going to have art. So on the basis of these 4 letters, we can have 5 different meanings using same letters, not even shuffling them back and forth, just deleting 1 out of 4 and then later keeping all 4 of them at once. So that kind of principle is what applies to reading and understanding encoded message from a single gene and later allowing it to be translated into different proteins. It's a little bit more complex than what I just said, but using this idea of CART and then playing with letters practically is exactly what's going on within a human body, except it is happening at a much higher, more sophisticated level. On the diagram itself, you see that there is promoter in the termination segment of the DNA, practically telling us, okay, well, this is part of the DNA, but it doesn't really code for any specific protein. It just rather tells us this is where you start. It's like indentation at the beginning of a paragraph, so you know it's not continuation of previous line of text, but rather it's a new paragraph. And then segment, which is called the terminator part, practically tell us where to stop transcribing so that the messenger RNA doesn't go in an endless form, but rather it has beginning, it has its end before it becomes allowed to exit the nucleus through the nuclear pore. All right, so a lower part of the picture in red, we have molecule of RNA, messenger RNA. And at the very bottom of the screen, we see it coming out of the nucleus through the nuclear pore. Let's find out what else is indicated or illustrated here. Just as we played with C, A, R, and T letters, we have parts of the messenger RNA that is divided into some useful parts, which we need for current synthesis of proteins. And we call those parts that will be used exons, where parts that are not coding for a particular amino acid or amino acids are termed introns, and they're cut, snipped out, and deleted. So splicing of 2 exons is happening here, while elimination of intron is exactly what we have done. If this is C, A, R, and T, we want to synthesize the word cat. So we're cutting out intron, carrying letter R, message, and we're putting these 2 together. So we don't want to say ca, and then long pause, T. We want it to be cat, and of course, these 2 ends of the messenger RNA have to be spliced together. I hope that this is kind of a little bit more entertaining than scientific approach, but as I stated a couple of times through a series of these videos, I don't plan to teach physiology here at some kind of very high academic sophisticated level. The principal aim of these videos is to help so many students grasp quickly and easily and understand the basics, which can be then used as a healthy and sufficient foundation on which you can keep building up and gaining the level of knowledge which you have to have in order to obtain passing grades in courses that you are currently taking. This illustrates interaction between small and large subunits of ribosomes, practically with a messenger RNA, And chain of green beads, of course, it's a chain of amino acids that are already put together, and we're successfully building the protein on the basis of messages encoded within the messenger RNA. A part of the diagram tells us components of the ribosome and their relationship to messenger RNA and a protein during translation. While from the other side, there are two interesting points that we want to see. One is that the large subunit does have A and a P site. And then on the small subunit, we have so-called the messenger RNA binding site. P and A site will be interesting for application on the next slide, where we're going to observe with a little bit more details as what's happening with process of translation and synthesis of a protein on the basis of encoded RNA, messenger RNA. A little bit unusual that number one doesn't start from the top of the screen, but we're gonna just find here what's going on. So this is messenger RNA, as you can see. Instead of any nucleotide carrying thymine, we're gonna have more of them with uracil. So this is a small subunit, which is already putting itself into place and looking for a start codon. What is meant by start codon? We have 4 letters, A, C, G, and U. And 3 letters are required to code for a specific amino acid. So it could be AAA, adenine, 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 containing nucleotides, or it could be adenine, uracil, uracil, or it could be cytosine, guanine, guanine, cytosine, guanine, cytosine, 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 cytosine. Basically, on the basis of these 4 letters, 4 nucleotides, we can create sufficient number of codes that would be more than enough for 20 amino acids, 10 of them essential, 10 of them non-essential. And we're going to have even surplus of extra codes that would not be coding for amino acids. So they could be used as the interpunction signs. They could be used to kind of capitalize the letters, saying not lowercase, uppercase. And that is what we have here as the start codon, which is essentially identifying first meaningful set of 3 nucleotides that would be matched by appropriate 3 nucleotides carried by molecule of transport ribonucleic acid. So what messenger RNA has as a set of 3, we call it codon. What is the matching set of 3 on the molecule of transport RNA? That is called the anticodon. And they have to match adenine with uracil, uracil with adenine, guanine with a cytosine, and that molecule of messenger RNA in interaction with the transport RNA is bringing first amino acid into place, making process of translation just started. On the diagram 2, large and small ribosomal subunits join and they form a functional ribosome, and initiator transport RNA fits into this P site. That is what we have already seen here, and first molecule of amino acid methionine is already in place. Remember, this is P site, this is A site. So basically, they are like some kind of spacers. When the first P site is occupied, A site is vacant. So what's going to happen is that ribosome will be going and screening messenger RNA 3 by 3 codons. And that is what we're going to see on the rest of this illustration, slide or segment number 3. Anticodon of incoming transport RNA pairs with the next messenger RNA codon at the A site. Okay, so at the A site, we have vacant space, And we have here uracil, adenine, guanine. So matching anticodon must be adenine, uracil, and cytosine. Adenine, uracil, cytosine. So good match. And this molecule of transport RNA is going to fit into A site. That's great. This is bringing second amino acid. And in the next process, amino acids on the transport RNA at the P site forms a peptide bond with another amino acid, which was brought in place by transport RNA at the A site. So we started with 1, now we have 2. Great. Segment number 5. First transport RNA, which brought first amino acid, the green colored methionine, is now released back into cytoplasm. If there would be more need for methionine amino acid, it can grab it and bring it back until building the protein. Transport RNA that was at the A site of larger subunit moves forward into P site and waits until another molecule of transport RNA does not land into A site, bringing the third amino acid, then fourth, fifth, and so on and so on. When the ribosome finally reaches the stop codon, that's usually a message which doesn't translate into any available anticodon carrying amino acid. So at that point, basically the ribosome and the very last molecule of transport RNA would know that the process of putting amino acids in chain is over and it would be released. Protein synthesis stops when the ribosome reaches the stop codon of messenger RNA. At the very bottom of the screen, you see here a bit longer chain of messenger RNA. And this is kind of summary of what top part of the screen identifies that messenger RNA allows a ribosome to simply glide on its surface, reading it code by code, bringing transport by transport messenger RNA molecule. And of course, it results in elongation and growing pattern of amino acid chain, which finally become complete and released into cytoplasm. Sometimes if a segment of messenger RNA is really, really long and it contains messages for multiple proteins, basically multiple ribosomes could attach simultaneously on it and keep synthesizing 2, 3, 5 different proteins, depending how complex the message is and what is the length of the messenger RNA. So we mentioned that the cell must divide, and in a typical division, which is called cellular mitosis, mother cell needs to split its content into 2 exact halves when it comes to nucleus and DNA, and 2 approximate halves when it comes to cellular membrane, cytosol, and organelles. Basically, any cell which divides does go through 4 predictable segments or events within a cycle, and they're termed prophase, metaphase, anaphase, and a telophase. They constitute mitosis. These words, please remember, are only applicable to events that are affecting and making changes within a nucleus. So practically prophase, metaphase, anaphase, telophase are just referring to sequences of nuclear division, not cellular division. Once the nucleus is divided, basically there is a completion of mitosis that is called cytokinesis. Through the cytokinesis, remaining cytoplasm, organelles, cellular membrane are given approximately half and half. So both daughter cells would get exact the same genetical content, and then perhaps plus minus 2, 3 more lysosomes to one other daughter cell. A little bit of a differences in of total volume, but it's insignificant because these are living cells. They would create enough organelles on their own. They will fill up the rest of the cytosol, whatever they needed, and then they would have full mature size. But this process essentially would involve a little bit of time. So in this pie chart, you realize that only this segment refers to mitosis, including cytokinesis, after which cell goes into interphase. Please don't feel that you have to remember these hour values because they are not exactly the same for each and every human cell. They're just approximate, but what is in the center part of the screen is also important, and that applies to all the cells. Two newly formed daughter cells obviously have to be smaller than the mother cell. Mother cell had 100%, and it gave 2 times 50% of its membrane, of its cytosol, of its organelles. So normally and naturally, you expect daughter cells to go through first growth phase, G1. Cells become metabolically active on their own, duplicate or multiply number of organelles, and compensate the rest of the cytosol until they grow up to appropriate size for being considered mature cell. Although they appear to be now mature, they have to go from G1 phase into S phase. S stands for synthesis, and during the S phase, the DNA that the nucleus of newly formed daughter cells contain must be duplicated. So each of the 46 chromosomes will have to be duplicated. So we're going to have 2 sister chromatids held by a centromere. That is happening during the S phase, synthesis of the DNA content. So we can say if at the end of mitosis, content of DNA was 100% in each of the 2 daughter cells, during the S phase, they build it up to the level of 200. So when they at one point split, generating new daughter cells on their own, they would have enough genetic material to give 100% to each of the 2 daughter cells. When the S phase is done and the DNA content is duplicated, cell must enter the G2 phase. So G1 S phase and G2 phases are part of interphase. Cell which doesn't divide is within its own interphase. A little bit of additional growth will be contemplated. Also some changes on the centriole, so it will start forming mitotic spindle. Some additional proteins will have to be synthesized, and the cell will become ready to become involved in its own mitotic cycle, going through prophase, metaphase, anaphase, telophase. And this cycle of life will continue for a number of different parts of our body for as long as we live. However, some cells at the end of their own mitosis and cytokinesis do not follow this path. They go into G0 phase. And at that point, they exit the cycle, which is not strange, neither it's rare. There are a number of cells that will lose their ability to keep dividing and generating new cells through the life. Example, which is easy to understand, are red blood cells. Why they can't multiply is because they get rid of their nucleus. That is what results in RBC, red blood cell, being in the shape of biconcave disc or donut, as sometimes people refer to it. Neurons, muscle cells also will lose in the first couple of months of our lives ability to multiply. So all of them will be within this G0 phase. Present, their numbers are kind of created sufficient for us to carry through the life. And for red blood cells, for example, we know there will be new ones that are coming from bone marrow, but those individual red blood cells would lose the ability to multiply on their own. So that is the cell cycle. And this explains as what's happening when DNA content must be duplicated. Top of the screen are two helices of the DNA, and of course, between compatible nucleotides having compatible nitrogenous bases, we still see hydrogen bonds. Once these bonds are broken, two strands are separated, each one containing compatible nucleotides. So this was guanine and cytosine. This is thymine and adenine, cytosine and guanine. And here was adenine that was separated from this thymine. But we know that in the process of building new strands, it will be built on exact the same basis, codes, and principles. So the new strand in a light green color will be practically matching the original second helix of the DNA, and the same would happen on the opposite side. So that result in two strands being separated, and then practically with copying, adding two new strands to form two sister chromatids carrying identical genetic material, from which they're going to be sent to two different daughter cells. The last one is just to take a quick look and try to spot some of these important things that are happening during mitosis. So we have here cell just before it starts dividing. It does have its own centriole, which started building more microtubules, forming mitotic spindle. Content of the nucleus, that would be chromatin, starts condensing upon itself. And in a prophase, of course, we start seeing DNA being fully condensed to form chromosomes, each of them being made of two sister chromatids held together at the centromere. For easier illustration, the illustrator opted just for two different chromosomes, not 46. So we can see one blue and one red with its exact duplicates that are still attached to it. In prophase, the cellular envelope kind of disintegrates itself, and in the meantime, mitotic spindle becomes quite fully formed, and individual microtubules that are part of the spindle will attach to kinetochore part of a chromosome. Early metaphase, the chromosomes will be lined up properly at what we call the equatorial plate. And mitotic spindle fibers or microtubules will be holding them quite firmly, so that in the next stage, anaphase, basically there would be separation between sister chromatids and shortening of the microtubules generated by mitotic spindle will start pulling apart sister chromatids. They're going to be pulled in the opposite direction. And in the late anaphase, we're going to start seeing formation of so-called a cleavage furrow, which is just a little indentation, identifying also that there will be some changes separating cytoplasm and cytoplasmic membrane of this mother cell into two individual cells. In a telophase, disappearance of mitotic spindle, reappearance of nuclear membrane, and further tightening of the cleavage furrow, identifying point of separation between these two daughter cells that will be practically fully separated, and they would enter their interphase. And then, of course, through cycle that's predictable, they're going to go into G1, S synthesis, and then G2 phase before they would become eligible and capable of repeating this process once again. I believe that we're just one slide before the end. Smooth muscle, nerve cell, red blood cells, we saw them entering their G0 phase, no further division. Sperm cell also would be generated on the basis of meiosis, not on the basis of mitosis, but it would also be unable to further divide. And epithelial cells that will likely stay capable of dividing and continuously regenerating parts of the body, which is either covered or lined up with epithelial cells. Another final view to the cell, which we have explored in these 3 relatively short videos. I hope that you have enjoyed it and that there will be some kind of benefits of going through these slides. So that would be for now, and see you soon. There isn't much more to say. New videos will be posted soon. I cannot promise whether it's going to be tomorrow or next week sometime, but we will be going further, and more anatomy and physiology videos will be found here on my YouTube channel. So stay tuned, and I hope that there will be something that you will find useful and interesting, and I hope enjoyable. Thank you very much for watching.